NADP is almost identical to NAD. If you notice at the bottom of the structure, there is an extra phosphate group on the 2' carbon of the bottom ribose. This phosphate is Mother Nature's way of telling the enzymes that the coenzyme should be used for the synthesis instead of for degradation. Like NAD, NADP also diffuses in and out of the active site. The structure of FAD is shown on this slide. Again, the AD stands for adenine dinucleotide, so we have two nitrogenous bases, adenosine at the bottom and a 3-ring system at the top. FAD is different in that the top ribose is in an open chain form instead of a ring form. This is a riboflavin derivative, and FAD is used for degradation, but unlike NAD, it acts as a prosthetic group to the enzyme. Another difference is that FAD can accept 2 hydrogen atoms, one on each of the 2 rings attached to nitrogen in each case, and they are shown here in magenta. Coenzymes are also needed for some of the transferases. I list here 6 of the coenzymes that are used for transferases. As we encounter these molecules during the semester, I may show the structure once, but past experience is that students don't particularly care for looking at the structures of these, and so I've gotten to the point of not spending a great deal of time on them. You should note, though, that biochemists do look at the structures for information. Finally, biotin is needed for carbon dioxide fixation. This is unusual in that the vitamin is used directly instead of being used as a vitamin D derivative. We next turn our attention to how we figure out things like whether or not something is a substrate, whether a coenzyme is needed, and similar types of questions. An assay is a way of measuring. If we have a reaction where a substrate is converted to product, then we could either measure the destruction of the substrate, in other words, the rate at which it disappears, or we could measure the rate of appearance of product. As a practical matter, we would pick whichever was easier to do. The components of the assay would obviously include enzyme plus substrate. We typically dissolve them in a buffer solution to maintain the pH. We would also have to add any required coenzymes or cofactors. To conduct an assay, what I would do is to mix together, for example, enzyme plus buffer plus coenzyme or second substrate if needed. In a second tube, I would have substrate. I would then incubate both tubes at the same temperature, usually 30 or 37 degrees Celsius, for a couple of minutes, which I refer to as pre-incubation, and then I would add the substrate to the other tube of enzyme plus coenzyme, etc. Depending on the reaction, I might be able to continuously monitor the reaction. For example, if NADH is produced in the reaction, I could have my spectrophotometer set at 340 nanometers and watch in real time for the appearance of the reduced coenzyme. Other times, what I would have to do is to withdraw an aliquot, perhaps once every minute, and then measure the amount of substrate or product manually. This would be referred to as a discontinuous assay. Biochemists use the expression unit of enzyme activity, and it is most often defined as the amount of enzyme that converts 1 micromole of substrate to product per minute under defined conditions of temperature, pH, etc. Recall that if A is being interconverted with B, that the rate of the forward reaction would be equal to K1 and the rate of the reverse reaction K minus 1. The rate is the ratio K1 divided by K minus 1. If we are measuring the destruction of substrate, our ratio would be a negative number. And if we were measuring the appearance of product, it would be a positive number. So these kinetic rules are all well and good, but one cannot actually go into the lab and measure rate constants. Instead, one is either measuring substrate or product. The most commonly used technique, or the most common technique used, was developed by Michaelis and Menton, who made the intellectual leap that if reactions were conducted for a short period of time, then there would not be sufficient time for the reaction to run in the reverse direction. This is referred to as steady-state kinetics. The rate constants are shown here. Theoretically, we could measure the amount of enzyme bound to substrate, ES. In practice, however, it is very difficult to measure, and therefore an assumption is to be made. The enzyme and substrate concentrations at time 0 can be known as it is the amount of material that we add to our assay tube. Steady state means that the rate of enzyme-substrate complex formation will be equal to the rate of enzyme-substrate complex destruction. Since the formation and destruction are known, we can come up with an equation shown on the third line of this slide. I will not walk through the proof of this, but we can do some algebra, some substitutions, and some assumptions to yield a workable equation. One example of an assumption that we make is that the reaction is run for a short enough period of time that less than 5% of substrate is converted to product. Since the enzyme can either bind substrate and convert it to product, or bind product and convert it to substrate, if less than 5% of the material is product, then the reverse reaction should not be able to occur. This is a key fundamental point of steady-state kinetics. The Michaelis-Menten equation, which should be committed to memory or tattooed on the inside of your eyeball so that you can see it when you close your eyes, is that the initial velocity is equal to the maximum velocity times the substrate concentration divided by the Michaelis constant plus the substrate concentration. This graph shows that as substrate concentration increases, the velocity also increases, initially very rapidly, and then plateaus off. The maximal velocity is the velocity at the plateau. So in this case, it would represent about 155 micromoles of substrate converted to product per minute. The Km is most often described as being the substrate concentration at 1/2 of the maximal velocity. Therefore, the Km would be the substrate concentration at which the velocity is just under 80 micromoles per minute, which would appear from this graph to be at about 100 micromoles of substrate. The shape of this graph is referred to as hyperbolic.